All right, so we are here to talk really quickly about checkpoints and what a checkpoint is. And let me see if I can make this go away a little bit. So a checkpoint is a point in the cell cycle in which the cell will stop and check some things out, check out its progress, make sure everything's going well, so that it doesn't get to the end of cell division and realize, oh, poop, we messed up somewhere, and we just got to just do apoptosis and die, because that's a waste. And so, what the cell wants to do is it wants to kind of check its progress at a few different places to make sure that everything's going as it should during cell division. So that's what a checkpoint is. At checkpoints, the cell's either going to get a stop, like something's wrong and we need to fix it, or something's so wrong we're going to abandon ship and do apoptosis, or a go signal. Everything's fine. You can proceed to the next step. And so that's how they regulate the cell cycle, is they make the cell stop and check things out. And if everything's good, then they continue. So when we look at the cell cycle, I did not draw this very big. All right, so we're going to get a big chunk of time. We have G1, S, G2. And then, just in the interest, and I'm not drawing these to scale at all, because prophase should be the longest of the cycle of mitosis. If we look at these three, these are interphase. These four are mitosis. And if you look at the one in the Nearpod or the Google Slides presentation, much better, much more to scale. So interphase would actually probably be all of this, because it's like 90%. But anyway, for our purposes for this, it works. All right, so we're going to have one checkpoint here, okay? And that is called the G1 checkpoint. And it's going to check a couple things. It's going to check for cell size, first of all, because if the cell's getting too big, if you remember back to surface area and volume ratio, if the cell's getting too big, got to divide. So it's going to check cell size and see, are we getting too big? Do we need to divide? It's also going to be kind of listening out for external signals. So if there's an injury, call them external factors or signals. If there's an injury, let's say you got a paper cut, then the cells on either side of that paper cut are going to get a signal to divide to fill in that paper cut. Sorry, there's the paper cut, to fill in that paper cut. And so that would be an example of an external factor or signal. So this is when they're going to be really listening for those signals. All right, so that's the main thing they're looking for there. If they say, hey, yep, we're too big, or oop, there is a signal, then they will go into cell division. And once they pass the G1 checkpoint, it will go to completion, unless something really bad happens and it goes to apoptosis. But it's not like, oh, the cell's going to hang out at G2 for a while. Once it passes this, it will go all the way until it gets back to G1. If it says, no, we don't really need to divide, it can actually jump out and go what's called the GO phase, where it's not dividing. That looks like the word go. It's not. It's G, like the number O. The number 0. So the G0 phase. All right. The second checkpoint is in G2. Now, if you remember, in the S phase, we copied our DNA. So in the G2 checkpoint, we are going to make sure that our DNA was copied correctly. Okay, this means it was copied fully. Okay, all 6 chromosomes, all of all 46 chromosomes were copied, and there's no mutations. And if there are any mutations, they'll go and fix it. If there's a lot of crazy mutations and it's just too much for the cell, then it will abandon this whole pathway and go into apoptosis. And the third and final is over here in metaphase. It's called the M checkpoint. And the M checkpoint is going to check to make sure that the kinetochore microtubules are attached on both sides of the chromosome. So what's going to happen is you're going to check and make sure, and if it's not, it's going to stop and give the cell time to fix that. Because what you don't want is you don't want it attached to one side and not the other, because then when this side starts pulling, if it's not attached here, this entire chromosome is going this way. And that's how we get things like Down syndrome. Well, this is mitosis, but in meiosis, that would be how you get things like Down syndrome or Edwards or Klinefelters, any of those trisomies or monosomies like Turner syndrome. So those are the 3 checkpoints. So I can describe the checkpoints. I can describe the GO phase. It's a non-dividing phase. Some cells go there forever. Some cells go there when they can hop back in as needed. And I can explain what a checkpoint is and why it's important. So I hope that helps. If you have any questions about checkpoints and how they help regulate, let me know.